Hi everyone, this is going to be the correction of the formative task that was set. In case you did not do it, make sure you complete it before you look at the correction. So the first part is describing the aim of the experiment. You need to say what you need to find out, how you're going to find it out, what and how. You should be able to understand the experiment clearly after reading this. You do not need to include any procedure here, but you want to say what you want to find and how are you going to find out briefly. So there's two information here, what and how. So if you read the question, this is the context. The context will always tell you what you're doing and how you're doing it. So it's talking about vitamin C in beverages. You are required to design an investigation to test for the presence of vitamin C. So this is the what you're doing. So to make it clear, I'm going to put it like this. And this information, I'm getting it directly in the context. This is what I'm doing. How I'm going to do that? Now this is going to be briefly about the method that you will use. So you know that vitamin C is tested using DCPIP. So then the how will be using DCPIP. So if you're writing the aim, these two things have to be present in your answer. What are you doing and how are you doing it? If you don't have any of these, please modify your response in your worksheet to add whatever is missing in your answer. So when we are going to grade your summatives, when I'm asking you to describe the aim, I will check that you've written what you're doing and how you're doing it. The second question is the variables. Independent, you see there's I in it. I. The I tells you what I change. And if you look at this topic here, this experiment here, what is the thing that I am going to change every time? One time I'm using milk. One time I'm using gross jelly, one time I'm using sports drink, one time I'm using orange juice. So every time I'm using a different beverage. Now when you're going to state the independent variable, do not just say I am changing beverages. Also state what are the beverages that you're using, because the question is describe. To be able to describe, you have to mention the name of the different things that you are using. And whenever we're looking at independent variable, there's always going to be several of them. There's only one independent variable, but there's different things that we are using, because I am changing it. So I'm using different items of that independent variable. There is only one independent variable. It's the beverages. That's the independent beverages. But I'm using different items of it. So I have milk, gross jelly, sports drink, and orange juice. So there's 4 items of that variable. Right? Now the dependent variable, dependent is what I'm going to measure, what I'm going to observe, what I'm going to measure in this case. When there is vitamin C present, the DCPIP, we saw that it's going to get decolorized. The color is gone. So observation is going to be the color change. This is what I'm observing. I'm going to check if the DCPIP turns colorless. And normally, if it turns colorless, we know that there is going to be vitamin C in it. So this is what I am observing. Usually, it's what you observe or what you measure. In this case, it's just observing color changes. Normally, for all the food tests, the dependent variable is the color change, because for all of them you're looking at how it's changing. For example, for the test for starch, it's iodine turning blue-black. For the test of proteins, it's the bio-red solution turning violet, etc. Then you have a control variable, what you need to keep the same to have a fair test. Now, if you look at this, we are using different beverages. Using different beverages. If I use a large amount of beverage, one is a large amount, one is a small amount, one is a medium amount, then my test is not going to be fair. It's just like if I'm giving a summative, I give one of you a difficult test, I give another one an easy test, so then it's not fair. Control, imagine I'm doing a summative. When I'm controlling the summative, all of you must get the same paper, the same level of difficulty, the same amount of time to do the paper. So similarly, control variable is what we are keeping the same to have a fair test. So I will keep the same volume of beverages. Do not say same amount of beverage. Amount is wrong. If I see amount in your summative paper, you will get penalized. Amount is not specific. You either say volume or define that specific quantity. Do not say amount. So I will use the same volume. I will use the same number of drops of DCPIP. And I will also set the solutions to the same temperature, because in the case of vitamin C, the temperature will affect how much of vitamin C is present. So normally at high temperatures, vitamin C or nutrients degrade. Many nutrients, they get damaged at high temperatures. So I'm going to keep all the solutions at the same temperature. So there's 3 control variables. Normally, if you give 2 or more, this is enough. Here I'm mentioning that all the beverages are at room temperature. So all of them are at the same temperature. Next, a hypothesis. What do you think the result would be if the beverage contains vitamin C? A hypothesis is, what do you think is the positive result? What do you think, or what do you think is the negative result? So normally, if it contains that food, if it contains that nutrient, what will happen? What will you see? So if the beverage decolorizes DCPIP, it means that it contains vitamin C. Normally, a hypothesis is given in the format if, then, because. But in this case, there's no because actually, so we are going to say if, then. So even in your summative, write it in the form if, then. So in this case, if the beverage contains vitamin C, then it is going to decolorize DCPIP. So this is the hypothesis. Now you need to have your method. Now in the method, there is 3 parts. There's the equipment, there is the diagram, there's the method. So these are the 3 parts. In the equipment list, I usually suggest that you write the equipment list as the last part, because you need to know what you're going to do in the method, and then you can cross-check with your equipment list to make sure that you have every equipment that you need for this test. Now, I want you to visualize what you used in the lab when we were doing the test. So if you still remember, we had our test tubes, we had our test tube rack. So the thing that you put your test tube in is a test tube rack. You had a dropper, and you had your solutions. Normally, the equipment list, you're going to have 2 parts. One is the equipment, and the next one is the chemicals that you used. chemicals or solution. Now the chemicals, it also includes the beverages. So you have all the beverages. You can list them. It would be good that you list the beverages, give the names of them. You will have your DCPIP solution. So to be able to write the equipment list, like the list of chemicals for all the food tests, you need to revise for your summative what are the chemicals that you need to do each of the food tests. Test for starch, test for protein, test for reducing sugar, test for vitamin C also. All of that you need to revise. And then the equipment list, from what we remember, you know that you had test tubes, you had test tube rack. It's the thing that you use to put the test tube in. You had beakers. The beakers were on the teacher table. This is in which you had the beverages in. So beakers to hold the beverages, and you had dropper to apply the DCPIP solution. Now I'm going to leave my list here, but as I'm writing the method, if I find that I need to add anything, I will come back to it. Now diagram, make sure that your diagram is not just drawing test tube, the test tube rack, the beaker and the dropper. That's not a diagram. The diagram, it should be a setup. It should be a setup of the practical. It should show me what is happening. So in this case, I can have my test tube, and I do not need to show all the beverages in my diagram. One is fine, just like I can see what is happening. So I have my test tube, I need to label it. And one thing very important is that the diagram must be in pencil. Please don't draw it in pen, because if you make mistake, you will have to use correction fluid, and that's not going to be nice. So use the test tube here, and then I'm going to add my beverage. So I can say about 1 ml of beverage. And then I had the dropper. That's the dropper. And then you can put a beaker with your DCPIP solution. And then you can show the drops of DCPIP being added to the beverage. So that is enough as my diagram. I do not need to show all the equipment. I do not need to show the test tube rack and all. This is fine. I am showing the setup, how the test is being done. Then for the method, I'm going to write the method. You can read it, check that you have everything from your answer. So this is my method. So add about 1 ml of beverage in the test tube. Add 2 drops of DCPIP. Observe if there is a color change. If vitamin C is present, it will turn colorless or it will decolorize DCPIP. Repeat steps 1 to 3 with each beverage. You see, I have not said take 4 test tubes in each of them, add milk, add grass jelly drink, etc. I explained for 1. So instead of saying 1 ml of beverage, you could say 1 ml of milk. Here, specific. I'm doing for 1. And then I say repeat with each beverage, and then you can name the beverages here. So you do for 1, and then you say repeat the steps with each beverage. And then this is optional. Record the results in an appropriate table. So this is what the table should look like. There's nothing for you to fill in. You don't know what the results will be, of course. So it's just showing how the results would be recorded. So make sure you have each of these steps in your answer. And then 2 safety precautions. Now the safety precautions relate to how you ensure that you do not, that you keep yourself safe actually. So To safety precaution, please do not give me general lab safety rules. Do not tell me do not run in the lab, no horseplay in the lab, for things like that. The safety precautions must be related to the chemicals that you're using or to the beverages that you're using. So the first precaution is that I should never drink any of those beverages. Do not consume the food samples. So I'm telling you what not to do and also why. Do not consume, as they might be contaminated. So there is the safety and there's the reason why this must be applied. Then the second safety, then I'm going to say, because the CPIP is a chemical, I will say do not allow the chemical to come in contact with the skin because it may irritate the skin. So it is a safety that is specifically related to my experiment. Now in this case, all the beverages were in the liquid state. Try to think, what would you do? How would the method be different if the beverage was in the solid state? Try to think about it. You will discuss about this in class.